is Harald Schmidt. I'm the coordinator of the Horizon 2020 project REPO trial, which went a little bit over 5 years, 5.5 years due to Corona, and where we started to change medicine by moving from the current symptom and organ-based disease definitions to mechanistic ones and exploit this by drug repurposing. That's where REPO stands for. But not only generate hypotheses or in silico trials, but actually prove this by conducting real phase 1 and phase 2 clinical trials. And I think we were quite successful. So let's dive into our scientific background and where we ended up. So one problem that we think we have in medicine in the moment is that we hardly understand any disease. What usually happens is that a patient with either no symptom or with a symptom goes to a doctor, and then the doctor maybe measures elevated blood pressure or reduced kidney function, reduced liver function, and then the disease is named after that symptom. So high blood pressure, hypertension, or heart failure, or kidney failure, or liver failure, or asthma, or all these descriptive disease terms. And because we do not know the cause of disease, so the real mechanism that caused the symptom, the only thing we can do, we treat symptoms. But if you don't know what's wrong, you can't repair it. And if you can't repair it, it stays chronically defect. So that's why we have so many chronic diseases. So we think simply the term chronic disease already is a capitulation of the current medicine. No criticism of the current doctors, but this is not how we can go on in medicine. We really need a paradigm shift. One example of that is drug therapy. That's what many patients experience when a doctor treats them. The doctor prescribes them a drug. So these were in 2015 the 10 drugs with the highest return on investment, so to say, but it's not different for any other drug. And the green patients are those which will benefit from those 10 drugs. But the red patients are those which will also receive those drugs. And we know from all clinical trials that they will not benefit from it. This is not a conspiracy theory. This is called the number needed to treat. Number needed to treat means how many patients do I need to treat with one drug so that one patient will benefit. So here, for instance, the number needed to treat is around 25, but there are drugs like antihypertensives or cholesterol-lowering drugs where these numbers can easily go 100, 200 or higher. The consequence of this, for instance, for the pharmaceutical industry was, if you don't understand diseases, you can only treat symptoms. And if the efficacy is so bad, how do you want to be better than the drug that's already on the market? And therefore, the success rate of the pharmaceutical industry gets worse and worse. It's called the better than the Beatles problem, or also Eroom's law. That's basically the reversal of Moore's law. Moore's law was what catalyzed the success of the IT industry, namely that we became better and better and in printing more and more transistors on the same area of a microchip. Therefore, the microchips became more and more powerful every 2 years. but actually in drug discovery, we get worse and worse and worse. So on the left axis, you see the number of new drugs approved per billion US dollar spent. And since the 1950s, this is going down and down and down. And mind you, that's a logarithmic scale. And this is not caused by tighter regulations after the thalidomide drug scandal. And it has also not improved considerably due to the introduction of biologicals, which some people think are wonder drugs. They have the same attrition rate as small molecules. So obviously, with the chronic diseases, with the inefficacy of drugs, and with this increasing failure of the pharmaceutical industry, there is obviously something fundamentally wrong how we define, diagnose, and treat diseases. Even for new drugs that make it to the market, or drugs that actually make it on the market, later we find out that 2/3 of the newly approved drug, when they are actually tested in real-world patient, not the hand-picked patient of a phase 3 regulatory trial, but the normal patient, who also have other diseases, who are maybe a little bit older than the ones in the trial or a little bit younger, then 2/3 of all new drugs have no benefit over the standard of care that was already there before. There are many, many things that go wrong at the moment, both in research, but also in clinical medicine. But let me focus here on one of the largest conceptual errors, namely that we have divided the human body organ by organ. For every organ, we have a clinical specialist, a clinical department, and a research discipline. So let's say here, neurology, the neurologist and neuroscience, cardiology, the cardiologist and cardiovascular research, and so on. So we really believe in the moment that just by looking at one organ, we can understand any disease. So anything we see there is the disease. And if the patient has any other symptom in any other organ, that must be a different disease. We give it a different name. We let it to be treated by different doctors and do research in another research discipline. Or even worse, we name a disease after the doctor who first described the symptom, like Parkinson or Alzheimer's. And as long as we keep on doing this, we will never discover molecular causes of disease. One good example why that is the case is if we look at rare diseases. These are diseases which are often caused by a single gene. So that's the exception. There we actually know the cause of the disease, a single gene. And these patients very often have symptoms in 2, 3, 4, or 5 organs. So this simple example tells you defining a disease by a symptom in an organ makes absolutely no sense. What's the alternative? The alternative is network medicine and big data. So basically looking at all the diseases and all disease genes at the same time. That's something impossible for the human brain to do. That's why in RepoTrial, we needed to collaborate with some of Germany's best bioinformaticians and innovative bioinformaticians who were also willing to listen to our medical needs, the precision that we need to make diagnostic and therapeutic decisions for a patient. And yeah, it worked out in the end. It, for me personally, started with this publication from Laszlo Barabazi, who is actually a physicist. And he works on all kinds of networks. He has written a fascinating book, Linked, that everything is connected with everything in this world. And here he worked on all human diseases. So these colored dots are the organ-based structure of medicine. So it's cancer, cardiovascular, ear, nose, throat, gastrointestinal, neurological, and so forth, as I've just shown you. And here are the diseases. And they are, in this case, linked through shared risk genes. So here, in the middle, for instance, there is a gene which is a risk gene in 3 diseases. So it links somehow those 3 diseases. And when he took all diseases and all risk gene or all shared risk genes at that time, he ended up with this network. And it's quite obvious that the network is not evenly distributed, but the diseases form little groups or clusters. And most of the clusters are multicolored, which means diseases with symptoms in different organs that we treat by different doctors and we do research on in different research disciplines actually belong together. They have the same risk genes. There are just different symptoms of the same mechanism in different organs. So basically, what we then do with our in silico trials is we reverse this, we flip it around. So what we now call the diseases, those colored dots, we now call just the symptoms or the phenotypes. And the mechanisms, the causes, we derive from the genes that link those diseases, that are the glue between those diseases in those clusters. However, just the genes is not enough. To exemplify this, we work on this cluster of diseases, which has at first sight a bit of a strange mix of diseases. One might think diabetes, obesity, asthma, heart failure, stroke, Alzheimer, Parkinson, is pretty wild mix. But all those diseases share risk genes. And one can look on network diseases also differently, namely by comorbidity. So comorbidity is if 2 diseases occur in the same patient again and again and again. Therefore, there may actually not be 2 diseases. There may be the same disease. That's why they occur so often in the same patients. Or 2 diseases may share the same symptoms. Then there may not be 2 diseases. There may be the same disease. That's why they have so many similar symptoms. So we can do these networks not only by linking diseases through genes, but also through symptoms or by comorbidity. And no matter how we looked at this cluster, the cluster is confirmed. Sometimes the genetics are a stronger link. Sometimes the symptoms are a stronger link. Sometimes comorbidity is a stronger link. But in principle, this is a cluster of common mechanism diseases. So we are not the only ones do this. There is a group of retinopathies that clinically look similar, but actually consists of 3 distinct molecular mechanisms. There's a group of primary immune diseases, which can, in this case, be linked through drugs that are active in more than one disease. That may also be an indicator that it's actually not 2 different diseases, but one disease. And here, all cancers have been analyzed genetically and can signaling pathways suggested with which all cancers can be described. So what we then did application-wise, once we constructed this disease cluster, we looked at the genes that glue them together. And from one set of genes, we could construct this signaling pathway. There's no need to go into the details yet. And then we looked in a clinical trial very closely at stroke patients here and at heart failure patients, two really high unmet medical need indication, in particularly a special form of heart failure. It's called heart failure with preserved ejection fraction. So the objective, measurable physical parameter of the heart, namely how much blood is ejected from the heart per heartbeat, is pretty normal. That's why it's called preserved ejection fraction. Yet the patients have heart failure. And none of the current drugs that work in other types of heart failure work in this type of heart failure. And it's about every second patient with heart failure that falls into this category. And stroke is, in most cases, when a blood vessel supplying the brain is clogged up. And then, of course, one can try to dissolve the thrombus, or one can try to mechanically remove it. But after that, there is no drug that can be used to treat a patient who had a stroke. So to protect the brain, for instance, from this damage. So we went into these two indications because they were all related to this signaling here. So what you then need to do is, of course, we don't want to treat all patients with heart failure, only those that have the symptom, plus where we can diagnose that this signaling module, as we call it, is actually affected. And this you will hear in later tutorials, how we actually come from the genes that link those diseases to a protein-protein interaction network and eventually to a signaling network. But then, in addition to finding drugs to treat any dysregulation of that, we need to develop a diagnostic to basically pick out the patients who have the symptom and the mechanism and treat only those patients with drugs that really correct this signaling pathway. For the other patients here in gray, we have to find other mechanisms. But if we treat those patients, we believe every patient will benefit, and no patient will be treated who will not have a benefit, as opposed to the figure that I showed you before on the high number needed to treat with our current disease definitions and our current drug approaches. So in this context, we have conducted a heart failure study and a stroke study. In the stroke study, the challenge was slightly different. There, the mechanism alters the signaling module in a slightly different way. But it's probably relevant for all stroke patients. However, there the limitation is that only during a certain time point after a stroke, so probably in the first 12 hours, it's relevant. So it makes no sense probably to treat a patient in the 13th or 14th hour after a stroke. Therefore, we identified, or we needed biomarker that determined this time window when a stroke treatment makes sense. And to speed up the process, we use drug repurposing. This is where REPO comes from. in this acronym for our project. And drug repurposing is basically cutting the process extremely short from, let's say, a basic science discovery to something from which patients benefit. Usually, the whole process to discover a new compound, test it preclinically, toxicology and so forth, and all the steps of clinical development can take 12 to 16 years. And it's extremely expensive. Drug repurposing takes drugs that are already on the market. So patients are already treated with those drugs, but for another indication. So you use those drugs for a new indication. And we thought in most cases, especially if the drugs are pretty safe and the indication is really a high unmet medical need, in those cases, the heart failure case and the stroke case, we could go straight to a phase 2 trial, which is in patients. Unfortunately, we had to, we're also required by the German regulators to do a phase 1 trial. Despite these compounds were on the market since many, many years. So there we had to find volunteers, healthy volunteers, to test patient or volunteer by volunteer whether any potential side effect may occur by combining those drugs, and only then could go into a phase 2 trial. And you will then later hear how these trials were designed, what the preclinical evidence was, and how the diagnostics and the drugs were selected, and how they actually work as a kind of role model, how we think in future, disease by disease, we could work up this process, look at the genes, design the disease or the signaling module that is affected in disease, develop a diagnostic to pick out the patients, find repurposable drugs to treat this signaling module, and yeah, then go as fast as possible into the clinic. The project was so successful that subsequently the EU awarded to us an even larger project over 25 million euros, together with colleagues from Switzerland. And that's called Repo4U. So here we are building a permanent European platform that will support anyone who is interested both in this big data approach to diseases, define diseases differently, and this validation of this by rapid drug repurposing. That has, of course, more aspects than just what I have described so far. You also have to think of, can I patent this? Is there a chance that a company will take it up and actually invest money then for maybe the subsequent later phase, phase 3 trials if necessary, the market entry, which all generates cost? Are there enough revenues? Because we're dealing with drugs that are already on the market, and very often they are not very expensive anymore. And now someone wants to invest money to actually bring them on the market. So that costs. Will those companies get their money back? Will it make sense for them financially? But also the regulatory aspects, patient aspects are important. What are the outcomes we should actually measure in such trials that are relevant for the patients, yeah? As opposed, for instance, this rather theoretical parameter of ejection fraction in heart failure. Maybe a heart failure patient has completely different parameters that are for him or her important to say, yeah, that is that therapy helped me. And sometimes people are a little bit surprised that it should be possible to do this drug repurposing, especially if we repurpose a drug from one target to another. So not for the protein, not for the disease the drug has been approved for, but also not for the target, so for the protein for which it has been approved for. And this is actually not a rare thing. So the axis here shows over 400 drugs that are approved in the US. And here on the left axis, it measures to how many proteins do these drugs actually bind. There are indeed a few drugs that bind only to 1 or 2 proteins. But looking here, there are some drugs that bind to hundreds of proteins. So in average, one drug binds to 32 other proteins with the same affinity and the same efficacy, if any effect derives from that. And this is due to the fact that drugs, also small molecules, need to bind to a pocket in a protein. So this pocket can be shallow or deeper, it can be hydrophobic, hydrophilic, but it needs to cover the small molecule, or the small molecule needs to have enough binding sites in here to actually stay bound with a high affinity and exert any effect. And apparently, the number of different drugs, different pockets on all our proteins is limited. The most frequent prediction that I have read is roughly 1,400. So there are only 1,400 different pockets on all our proteins. And that would mean if we have discovered 1,400 drugs that bind to different pockets, that's it. The 1,400 first drug will bind to a pocket for which we have already a drug. So one thing is clear, we will not need to eternally look for new drugs. So drug discovery will come to an end, which is another argument that there is a huge potential for drug repurposing. If, for instance, the pocket that we need to address on one protein is already addressed on another protein, so we then just move from that protein to this protein and reuse the drug. Yeah, with this, I would like to conclude and really suggest that you also listen to the talks by my colleagues, for instance, by Ana Casas and Christian Nogales. Also have a look at the NetREX platform, which is a combination of multi-scale bioinformatics to integrate, as I said, disease-gene relations, disease-symptom relations, comorbidity, and so forth. And by this, come to a conclusion which genes are worthwhile to pursue. And then you will hear from Christian how to construct these disease modules and these diagnostics, and from Ana Casas how you then exploit this pharmacologically. So by network pharmacology. Since these disease mechanisms are little networks, we also need to treat networks, and that's not complicated, but it has huge advantage over another dogma that we will also overcome, namely that one disease can be explained by one protein or one target and treated with one drug. That's not possible. the biology and the biology of diseases is a little bit more complicated than that. As I said, little networks which need a little bit more tricky diagnostics and a little bit more sophisticated drug therapy, but then with high precision and high efficacy. And we're very happy that REPO trial was so successful and that we have the opportunity with the follow-up project REPO4U to generate something for Europe and yeah, everyone interested in any aspects, maybe from basic science, maybe from patient support groups, from regulators, to make this new way of medicine really become reality for patient benefit as soon as possible. Thanks for your attention.